everyone. Thank you all for joining us for our February Research Spotlight webinar. I'm Dr. Sarah Hernandez, the Director of Research Programs for the Hereditary Disease Foundation. I want to start by thanking everyone who donates to support the HDF. A massive 82% of our budget is spent supporting research. Because of you, we're able to fund the exciting research that you'll hear about today, and we're also able to have these webinars to tell you about that research. So thank you. Today's webinar covers an exciting topic that I know a lot of you are interested in, and that's CRISPR. You may have heard that this technique is transforming the way that some diseases are being treated, like sickle cell disease. And you'll hear today that it's also being used for HD research in mice to explore potential new treatments. Today, we're going to hear from a team that was awarded an HDF Transformative Research Award in 2023 for a project titled Therapeutic Targeting of Somatic CEG Expansions with Precise CRISPR-Based Editing. So this award was for $1 million given over 2 years. And the goal of this project is to develop genome editing therapeutics that target the cause of the CEG repeat expansion in Huntington's disease. So if successful, this work will lead to preclinical studies that test the effect of altering genes that control CEG expansion on the behavior of mice that model HD. We are joined today by the lead PI from that award, Dr. Ricardo Mouro Pinto, and we'll be joined by other members of the team for the Q&A session of the talk. So Dr. Zach McLean and Zanetta Matuszek will join us. Dr. Mouro Pinto is an instructor in neurology at Harvard Medical School, Mass General Hospital. He's interested in human genetics and neurosciences with a strong research focus on repeat expansion associated disorders, such as Huntington's disease and Friedreich's ataxia, particularly concerning mechanisms of trinucleotide repeat instability. Dr. Zach McLean is a postdoctoral research fellow at the Center for Genomic Medicine at Massachusetts General Hospital, Harvard Medical School, and the Broad Institute. He works in the lab of Professor Jim Gisela, where his research focuses on in vitro modeling of CEG repeat instability and genetic modifiers of Huntington's disease. Zach was awarded with an HDF postdoctoral fellowship in 2021 for his work on modifiers in HD, and he's an active member of HDF's Young Investigator community. Zanetta Matuszek is a PhD student in the Department of Molecular and Cellular Biology at Harvard University and a member of Dr. David Liu's lab at the Broad Institute of MIT and Harvard. Zanetta's research focuses on the therapeutic applications of CRISPR-Cas9-based genome editing technologies, especially for trinucleotide repeat disorders like HD. We are very excited to have you all with us today, so thank you so much for joining us. Before we start, I want to remind our audience that you can ask questions in the Q&A box throughout the talk. So Q&A box, not the chat box. Feel free to ask both technical and non-technical questions. We should have about 20 minutes at the end for Q&A with the speakers. So send your questions in as you think of them so that you don't forget, and we will address as many of those as we can. I also want to let everyone know that this will be recorded and will be available on our website and YouTube channel in about a week or two. So if you want to watch it again or if you want to share it with someone, it will be available. Thanks again to everyone for being here, and I will pass it over to Ricardo.
Thank you, Sarah, and thank you everyone for the invitation to present our work and for the award for our research. So Sarah led me to believe that you'd be interested in learning a little bit about me. So here we go. So originally from Portugal, grew up in Porto, and then went to pursue my higher education in the UK. So did a bachelor's in genetics in Cardiff University, and then moved to London to study human molecular genetics, and then did a PhD in neuroscience in the lab of Dr. Mark Pook, working on Friedreich's ataxia. And this is really where I became interested in repeat expansion disorders, and this was my first contact with Huntington's disease. During my PhD, I became very much interested in repeat instability, and that led me to pursue a postdoc in Boston in the lab of Dr. Vanessa Wheeler, who was a leader and one of the only PIs at the time working on somatic instability and Huntington's disease. So this has been my scientific path. Now run a small lab here at MGH with generous support from foundations, the NIH, and industry. So I'll be brief in terms of the introduction about Huntington's disease, but I'll just touch on a few key points. The fact that this is an autosomal dominant the genetic disorder, and it's solely caused by an expansion in the CHE a CHE repeat in the Huntington gene, and individuals who inherit approximately 40 or more CAG repeats will end up developing Huntington's disease throughout the course of their life. And it just leads to primarily neuronal degeneration. And as you can appreciate here in the patient brain, you end up with a significant amount of neuronal loss, particularly in the striatum. Another aspect that is important to remember ourselves is that the longer the repeat, the earlier the onset of the disease. So there's a strong relationship between the size of the mutation and the onset and the significance of the symptoms. And this can affect this can manifest in a variety of ways, both cognitive, motor, and behavioral. I'll touch on a couple of aspects that are particularly interesting in repeat expansion disorders. And one common aspect of Huntington's disease is anticipation, meaning that individuals in subsequent generations will end up developing the disease earlier than their parents. And this is primarily caused due to intergenerational instability. So the repeat changing in size from the parent to the child. And it primarily occurs when it's transmitted from the father. So you can appreciate here when the repeat is transmitted from the mother, so that would be the size of the repeat in the mother, and the size in the child tends to be more or less the same. There's some fluctuation, but it tends to be more or less the same. On the other hand, when that transmission is done from a father, the child tends to inherit a larger CAG repeat, which will contribute to an earlier onset. So this is what we refer to as intergenerational instability. Another important aspect is what we call somatic instability, which is the repeat can expand from its starting size, the inherited size, in a time-dependent and in a cell type specific manner. So these are post-mortem brain samples from a patient who died before approximately 13 years before predicted onset. This individual inherited a CAG of 41, but in certain neurons in the striatum, this repeat has expanded all the way up to the hundreds, even as high as 1,000. And you can appreciate here that with time, this actually starts occurring not only in the striatum, but in a variety of other regions in the brain. And the reduced level that you might see in the striatum at this point is primarily related with the fact that at this point, the striatum is mostly lost. We've performed some analysis, and I won't bore you with too many details here, but essentially trying to understand if somatic instability, where does it occur, and is it shared across patients? And what we can see is that the propensity for some tissues to expand is shared across patients, and it's primarily in neuronal tissues, while in the periphery, you don't seem to have that much expansion. So here I'm just color coding and ranking the types of tissues that are potentially accessible by a biopsy, where there's little instability, but in certain regions of primarily in the CNS, you have more propensity for the CAG to expand. And I'll just touch on a couple of studies. So this is work led by Nat Hines' lab at the Rockefeller, and this was just recently published, and this is from Stephen McCarroll's lab here at the Broad. And these are really important pieces of data that show that the CAG expansion occurs only in certain cell types within the striatum. So you have a variety of different cells that where the CAG repeat stays at around the size that it one is inherited. Then in medium spiny neurons, which are the main neurons that are affected in the striatum of a patient, the repeat tends to expand. And here we're looking at a case with 44 CAGs, a 54-year-old female. But here Steve is actually showing that a similar finding in a variety of other patients, where you can see that those expansions occur only in the medium spiny neurons and not in other cells in the striatum, suggesting that these affects the vulnerability of these neurons. As I sort of alluded to, there's this relationship between length and age of onset. However, it's not a perfect line, and there are individuals that will develop the disease much earlier than expected, and others that develop the disease later than it was predicted solely by their CAG repeat. And in a study performed by Vanessa Wheeler, she looked into the instability in the cortex of these individuals. So taking individuals sitting at this extreme and that extreme, so either earlier than expected or later than expected, comparable CAGs. And what you can see is that the individuals that develop the disease earlier tend to have a higher degree of somatic expansions. So these would be the allele in gray, the allele that is inherited, and all of these would be expansions. And you see primarily these very large expansions happening in individuals that have an earlier onset. And this is sort of another representation of the same data, showing that in general, the individuals with earlier onset have a higher frequency of these really large expansions. Another important study that was conducted recently was a GWAS, or a genome-wide association study. And I'll try to explain these simplistic terms, but essentially it's taking those individuals that deviate from the predicted onset and trying to understand if there are polymorphisms in their DNA, other genes that are sort of associated with an earlier or later onset than expected. These studies sort of identified a number of different genes. Some of them we didn't know what they were doing. Some of them we had already studied in mice, and we had already known that they were modifiers of instability. But we still didn't have a full picture of what was going on and how these so-called modifier genes that were identified operated. As part of a study funded by the Huntington's Disease Society of America, Berman Topper Fellowship that I received, we designed a study to test how these candidate genetic modifiers operated in the context of an HD mouse model and how they potentially affected somatic expansions. So we've used CRISPR in this study, so not really in a therapeutic setting, but in a lab testing way. We designed guides against genes of interest. We deliver them to the animals and turn off genes of interest, and then age them and sort of evaluate the impact it might have on instability. So if there's no effect, it's going to look like that. If we turn off a gene that is protective, we'll sort of accelerate the expansion process, and we'll see more expansions. On the other hand, if we turn off a gene that is promoting expansions, we're going to slow this down. And in fact, we can do that, so we can see that this is a phenotype, or this somatic instability increases in mice with age. So as the animal ages, there's more and more CAG expansions. But when we turn off some genes of interest, we can completely stabilize the repeat. So these animals are the same age as these animals, and we completely stabilize the repeat. As you can appreciate here, these are just some examples of what this data sort of looks like. With aging, you get more of these expansions. Turning off, for example, MSH3, you completely stabilize it. Turning off FAN1, you accelerate that process. So we expanded this study and we really screened a large number of genes. And I won't go into too many details about what we found that to be negative, but highlight primarily the positive things. So genes that are landing on this upper panel would be things that accelerated the rate of instability in our screen. So they're essentially protective genes. And we identified FAN1, MSH6, and PMS2 as the strongest suppressors. And we also identified another set of mismatch repair genes that when knocked out, you completely stabilize the repeat. So these are sort of the genes that are bad. There are the genes that are promoting CAG expansions in patients and in mice. So we know that when we get rid of them, we prevent CAG expansions. But what's important to take from here is that this is essentially one DNA repair pathway. We know what to target now, and we know that this is primarily the mechanism that where we need to intervene. But I'll sort of touch a little bit on how this DNA repair pathway is comprised. So there's sort of two different components or dimers of repair that they interact in different ways, and some are shared, others are sort of unique. They only have one partner. And these are primarily involved in the recognition, and these are then involved in the repair. So the recognition of the mutation, in this case, it will be a CAG slip out, a CAG. Whenever you have an expanded CAG, it tends to form a different structure that looks odd to the DNA repair machinery, and it's recognized by it, and then it tries to repair. So here I'm just trying to summarize what happens in HD when you turn them off. When you turn off any of these genes, which are the ones that have a beneficial effect. So the downwards arrow will be slowing down CAG expansions, while the green arrow is they're accelerating, so they're bad. But these are sort of in terms of how you'd go about with a therapeutic, in terms of intervening and inhibiting these genes. Another important aspect is that HD is part of a group of other repeat expansion diseases. If we're thinking of developing a therapeutic, we might want to try and understand how this works across a variety of different diseases and potentially develop a drug that would be applicable not only for HD and CAG repeats, but also other diseases like Friedreich's ataxia that is caused by a GA repeat, or Fragile X with a CGG repeat, or potentially spinocerebellar ataxias, which are also caused by a CAG repeat. And what we see is that the results aren't entirely the same across these different loci. Another important aspect is that some of these are associated with cancer. Mutations in some of these genes are associated with cancer. So one would need to be careful in designing drugs to modulate the levels of these genes. But this is sort of identified three top candidates for therapeutic development in this space, and that would be MSH3, PMS1, and MLH3. And there are significant efforts going on from academic labs and from pharmaceutical industry in terms of pursuing some of these targets for Huntington's disease. I'll just touch on some of the studies that we performed for MLH3. We didn't know exactly what MLH3 was doing in the context of Huntington's disease. We know it has an endonuclease domain, and that's essentially a scissors component to this protein. It has the ability to go to DNA and cut it. So we knew that MLH3 was involved, but we didn't know exactly how. So we're trying to understand if this scissors component was important for the CAG expansion process. And we used mice. We used mice that have the scissors component and mice that don't have those scissors being expressed. And I'll just focus on some of these pieces of data, where here in a normal HD mouse, you have a high degree of CAG expansions in the striatum. But in a mouse that is missing the scissors, so it's still expressing the protein, but it doesn't have the ability to cut DNA, you completely stabilize the repeat. And this is maintained over time and across tissues. So we know that the scissors component of this protein is essential for promoting CAG expansions. We started thinking about how could we target this therapeutically. And in collaboration with Ed Grabzik at the LSU, who was developing ASOs targeting the endonuclease domain of this gene. So the human MLH3 can be expressed in two flavors, one that contains the scissors and another one that completely skips the exon that expresses those scissors. So that will be a shorter variant. And Dr. Grabzik had shown that this was important for GAA expansions. Whenever you use this drug, it could slow down the degree of GAA expansions gained. So here I'm sort of summarizing how that looks like, and here actually showing that in humans, variant 2, so the shorter variant, the variant without the scissors component, is normally expressed. So in fact, in some tissues, it's expressed even more than the variant with the scissors. So we're confident that developing a therapeutic that promotes expression from one variant to the other, rather than simply turning it off, would be better tolerated. We would be promoting the expression of something that is already present in those cells, but that it is not associated with CAG expansions. So we've collaborated with that, with exploring that ASO strategy. We've done it in HD patient fibroblasts. So we had to treat this on a regular. So these are cells that are taken from a patient. They grow on a dish, and they actually demonstrate, they show CAG expansions with time in a dish. So we treated them with this cocktail of ASOs on a regular basis, total of 6 weeks. And we noticed that we were affecting the splicing of this gene in the way we wanted. We were promoting the expression of the variant that is lacking the scissors component. And when we did so, we stabilized the repeat. We slowed down the CAG expansion. We've also tested this in mice with tailbone injection. So it was sort of a proof of concept. We wanted to make sure that this was also the case in mice. We had to do a very intensive treatment regimen with 3 weekly doses, because the ASOs were being cleared too effectively by the mice. But even though it wasn't as successful as we thought, primarily in terms of limitations associated with ASOs and this particular chemistry, but we could see that we were seeing a suppression of CAG expansions in the tissues where we were reaching with this drug. And that was here in the kidney, where you had normally these many CAG expansions that you completely inhibited. But the difficulty we faced with ASOs and with this particular therapeutic modality made us start thinking about how could we do this with CRISPR. And I'll just very briefly touch on how CRISPR came about. So this is a system that is present in bacteria. This is sort of their bacteria's immune system. They when bacteria get exposed to a virus, pieces of the RNA or DNA from that virus get incorporated into the bacterial genome. And then when the bacteria is exposed to the same virus, it will have the ability to express a little bit of that RNA that was from the virus and use the Cas enzyme to go and destroy the viral genome that is being in the new infection. So this was sort of adapted. This is limited to bacteria, and this was sort of adapted to human cells. As the Nobel Prize with Jerry. But so here is sort of showing another diagram of how this was adapted to work in human cells, where you're going to have genomic DNA, you're going to express Cas9 with a guide that is an exact match to your sequence of interest in the genome. And then the Cas9, this enzyme, will create a double cut, will chop this up if it recognizes both that sequence and an important component, which is a PAM sequence, which is a protospacer adjacent motif, which is about 2 to 6 bases. If you have both these and that sequence match, Cas9 will cut. And then there are a variety of different ways where you can adapt this to either turn off a gene, sort of destroy it, mess up with its sequence, or use it to then incorporate a sequence of interest. But I won't get into too much detail on that because this is sort of has risks associated with it. Introducing double-strand breaks it has its own challenges and risks. And so others have been working in the field to sort of engineer Cas9 enzymes, CRISPR enzymes, to do slightly different functions with a little bit more control and a bit more precision. And some of this work is led by David Liu at the Broad, who invented both prime editing and base editing. So I'll touch a little bit on what base editors can do. So essentially, these enzymes have been modified so that they no longer do double cuts. They only do what we call a nick. So they only cut on one side, but they've also been fused to deaminases. And here I'm showing an example of what we call an adenine base editor. So essentially, you control it the same way with a guide to the region of interest. And then this deaminase will recognize the A and modify it so that you can change A's to G's or T's on the other side to C's. So this is limited. An adenine base editor is limited to modifying the A. The cytosine base editor, on the other hand, will do the similar thing on the cytosine, on the C. So you can change C's to T's and conversely G's to A's. So essentially you can do all possible kinds of base modifications without breaking up the DNA. So essentially you just nick it once and then it's repaired and filled in using the new base that was modified. So you'll have a permanent single base change So you started with an AT and you end up with a CG. Another aspect that is limiting of CRISPR, and I've alluded a little bit to that, is the PAM sequence, which normally in wild type Cas9, it's an NGG. But that sort of limits your ability to target the genome. There's so many places in the genome where you're going to find that sequence. And work led by Ben Kleinsteiber, who's a colleague here at MGH, he's engineered this Cas9 enzyme in a way that it can recognize a variety of other PAM sequences. And theoretically, you can sort of target the entire genome with this. So we've sort of incorporated both these technologies into our efforts for targeting MLH3 for CAG expansions. How we thought about it, one important aspect is that splicing, this is sort of a complicated process to explain, but essentially there's the splicing machinery will recognize a certain sequence and which will tell it, okay, let's start incorporating this piece of the gene, which is the one that contains the Caesars. And this is a very conserved sequence. If we look across species, human, fish, fly, fungus, all the way, we have this AG motif immediately before the exon starts. So we hypothesized that if we were to change either the A or the G in this region, we would be able to disrupt this splice acceptor and skip it. So essentially do the same thing we were doing with the ASOs, but promote the expression of the flavor without the scissors. So we've collaborated with Ben Kleinsteiber for doing this. We essentially designed guides across, all possible guides across this region. Guides that would work with an ABE and guides that would work with the CBE. So either targeting that A or targeting that C. So we're thinking of disrupting either one of them. And what we've done, this is sort of some of our preliminary results. I'll just this would be sort of the results you'd obtain with a wild type variant. So wild type generic canonical PAM variant, we wouldn't have seen much of an effect. But when we use these modified ABEs, we found combinations that are rather effective at modifying one of these bases of interest. And so we've been pursuing this validation. What we've shown as well is that when you make, so in every cell you have 2 copies of the gene. When we edit just one of the copies, we shift the expression of the isoform with endonuclease to the isoform without the endonuclease. So we reduce the bad isoform. But when we actually edit both copies in the cell, we completely eliminate the bad version of MLH3. So we're excited about this, and we've actually done some preliminary studies in terms of how does this affect instability. And here I'm showing data from the cell line that Zach developed, where with time, the CAG tends to expand. So this would be around 120 CAGs, and it's gaining repeats as you keep it in culture. On the other hand, when we have these both alleles edited with our base editor, we completely stabilize the repeat. So it doesn't change. It doesn't grow all the way up to 4 weeks. It didn't increase its CAG. So we're very excited about this. This sort of leads us to acknowledge the tremendous honor that it is of receiving the HDF Transformative Research Award to pursue these sort of lead CRISPR base editors. There are ongoing efforts that include testing these reagents in mouse models. So one thing we've had to do is that since these sequences don't exist precisely the same way in mouse, we had to sort of humanize this mouse so that we can test reagents that we would eventually use in patients. And another aspect is we need to test these reagents in a variety of different patient-derived cellular models. So that's ongoing, and that's the work we've got planned for the next couple of years. There are two important considerations to take note of. One will be safety. We need to make sure that these, even though they are making the desired edits, we need to make sure that they're also not making undesired edits, what we call off targets. So that will be a strong effort. We're going to pay close attention to those, to that aspect. The other aspect is delivery. How do you get these reagents to the medium spiny neurons in the brain of a patient? And that is also a considerable aspect that we're going to explore. We're going to start primarily with viral deliveries, but we're also going to consider lipid nanoparticles or extracellular vesicles, which are sort of different vehicles for taking our drug of interest. And I'll sort of acknowledge the team and the variety of different skills. So we've got Jim Gazella, who's working in the field of HD and for the last 40 years. We have David Liu from the Broad, who's invented both prime editing and base editing. We have Ben Kleinsteiber, who has engineered these PEM variants here at MGH. And we have Ben Deverman, who is pioneering these viral delivery methods. So these viruses that are capable of crossing the blood-brain barrier, and so you'll administer them systemically, and they will still reach to the brain. We have Kat Lutz at the JAX, where they have a tremendous amount of expertise with preclinical studies. Joe Navan at Vesigen, and this is an industry partner. They're developing extracellular vesicles as an alternative way of delivering CRISPR reagents. And Vanessa Willer here at MGH, who's my mentor and an expert in somatic instability. So with that, I'll just sort of summarize more or less our therapeutic strategy, how we see this. So an individual will be born with an HD patient will be born with a certain CAG size. And throughout its life course, certain cells, certain neurons will start expanding with time. At a certain point, they will expand beyond a certain threshold that will lead that cell to die. That neuron will die once it expands too high. And that will be a progressive aspect of the disease. And we're marking here, for example, onset at this point. An individual who has an onset delaying modifier, so has some genetic variants that reduce the rate of somatic expansion. So the repeats will expand slower and slower. Therefore, they will reach that threshold at a later time point, and they will have less neuronal loss, and therefore a later onset. On the other hand, an individual that inherits genetic variants that are accelerating the rate that the CAG expansion, the cells will expand faster, will get to that threshold earlier. They will start dying at an earlier time point, and the patient will develop symptoms earlier than predicted. And our hypothesis is that if we were to develop a drug that intervenes early and prevents the CAGs from expanding, that you would sort of press pause on these mechanisms, preserve the neurons below that threshold, and ideally delay the onset beyond the normal lifespan. Again, sort of acknowledge everyone in the lab. The majority of this work was led by J.D. Roy, who was an HDF fellow, and a number of our collaborators and our funders. Postdoc positions available. If anyone is interested, please feel free to reach out. I'll thank HDF for the opportunity to present, and just want to introduce some of my colleagues here for the Q&A. Zach McLean in the Gazella Labs, an HDF fellow, who's been developing a lot of these cellular models for driving somatic instability. And Janetta, who is a PhD student in David Liu Lab, who's working with a lot of these CRISPR reagents that we're talking about. So looking forward to discussion, questions.
Thanks, Ricardo. That was fabulous. Thanks so much for sharing the work and introducing Janetta and Zach. So thank you to everyone who has been submitting your questions. Keep submitting those throughout the Q&A. We'll try to get to as many as we can. Again, technical, non-technical, just put them in the Q&A box and we'll get to as many as we can. Okay, so some that have come in are really related to timing. When do you think a CRISPR type treatment will be available for HD? And a few people specifically asked, do you think it would happen in 10 years or less, or within 10 years? So I don't know if you can opine upon that.
There are a variety of different CRISPR approaches for treating Huntington's disease. I've just presented one, that is the one we're exploring. I'm sure there are others that are being explored, and some of them might be even further in development. It's hard to predict timelines for this, especially when industry is still going through that for the first time. They're still understanding exactly, going through conversations with regulators. What is it exactly that is needed for these different trials? What sort of different safety precautions everyone needs to provide before initiating these trials? But the fact that so many industry companies have sort of presented positive results and positive indications from the FDA, and they're moving along with some of their pipelines, granted for diseases that are a little bit more accessible than neurodegenerative disorders. But I think we're going to have a better roadmap as soon as some of these drugs get approved.
Thanks, Ricardo. So there's some questions around why you're targeting somatic instability associated genes rather than Huntington. So Pinar Akakaya, apologies if I butchered that, says, very nice talk. Why would you go for the splicing acceptor modulation instead of removing the disease-causing CAG repeats with CRISPR?
I think I think one effort does not eliminate another. I think there are for sure many groups working on that strategy. They don't come without their own challenges. So what we believe with this strategy is that it should be safer, should be easier, and it has the added advantage that it might be applicable not only to HD, but a variety of different repeat expansion diseases. Thinking of the spinocerebellar ataxias, myotonic dystrophy, Friedreich ataxia. So that is one of the rationales for going after a modifier, going after a DNA repair gene that affects the rate of somatic expansions.
Thanks, Ricardo. So maybe here's one on instability. So maybe we can bring Zach in here. So from Beth Hoffman, do we understand what sets off the expansion, or does this occur stochastically from birth?
So this is something that we're still trying to understand. We know that you require certain things to initiate this somatic instability. This is one that you get transcription going through the repeat, that it's a thing that's being turned on. And this is then enabling structures to form that have then come and repaired by these things Ricardo was talking about. And so like Ricardo mentioned, we know that if you remove some of those key enzymes that come and try to repair this, that do a bad job, that you can then stop this repeat from expanding. So I guess that's a little bit of the mechanism.
Thanks, Zach. So maybe we'll bring Janetta in here. So Ryan Van Dyke asked, what are the biggest challenges to get CRISPR into the brain?
So I think currently the biggest challenge is delivery, especially in terms of treating patients. So we are able to deliver CRISPR reagents in the brain and to other tissues in mouse models. However, those delivery modalities might not be perfect for using in patients. And that's why I think this area has been very widely explored recently, and majority of the clinical trials right now, or even recently the FDA approved CRISPR-based drug, are drugs that are targeting blood disorders or like other liver disorders that can be targeted with like delivery modalities that are maybe slightly safer than AAV. So I think the delivery is now the biggest challenge for targeting the brain in terms of CRISPR.
Thanks, Janetta. So I think we'll go back to Ricardo for this one. This is from an anonymous attendee, says, amazing work, congrats. Does genetic manipulation of these modifier genes affect symptoms in these mice?
So we haven't done, so that's actually some of the work that we've got planned. We haven't done a lot of that investigation. We do know that we change some of the downstream phenotypes of the HD mutation. So we know that we change the amount of mutant protein that gets accumulated in the nucleus when we slow down these CAG expansions, but we haven't done a lot of functional studies in terms of behavior. It's been challenging, and I think we're at a point where we're starting to learn exactly what might be the best model for doing that. It's been challenging to find models with the right CAG size, and what is that correct model to stop CAG expansion and still have an effect. We don't want to go with models that are where the repeat is already too large, that clearly have a phenotype, but if you stop the somatic CAG expansion, you're not going to do anything because that CAG repeat is already too large. We've been operating more on the safer side with smaller CAG sizes, with the 111 to 120 CAGs, but these mice don't really have that much in terms of symptoms, of motor symptoms. So it seems like we're trying to understand now exactly what would be the right CAG sizing in mice. There are studies not done by ourselves, but I'm aware of that you can modify motor symptoms and transcriptional signatures and a variety of other phenotypes in a mouse when it's around 140 CAGs. So if you stop CAG expansions at around 140, you'll have a therapeutic impact in that mouse model.
So this is kind of related maybe to the next question that I want to address or direct towards Zach. So this is from Eva. Thanks for the amazing talk. I would love to ask, to which extent is cell death in HD attributed to CAG expansions? I know you just mentioned the 140, Ricardo, but maybe you could add to that, Zach.
So I guess this is related to like the underlying model that we're working from, that we've learned from like these extremely large human genetic studies, that like first you need a significant amount of repeat expansion. And then once you reach a certain threshold, that will result in the neurons in the brain dying. So we're still trying to understand the second component of it, of what the threshold is, of how long the repeat needs to be before it results in the cells dying. But this is a area that a lot of people are working on, and like Ricardo mentioned, our colleagues in the Broad Institute, Steve McCarroll, they have a lot of new exciting research where they're analyzing these HD patient brains at the single cell level to try to uncover these critical thresholds.
Thanks. So going back to Janetta, Igor asks, does CRISPR treatment pass through generations?
Oh, that's a great question. So there's been a big debate about germline editing regarding CRISPR. And I think as of now, as far as I'm concerned, this is not the case. So we are not editing germline, so it would not pass the generations.
Thanks, Janetta. So back to Zach for this one. This is from an anonymous attendee. Thank you all for your dedication and work in this area. Can you discuss more specifics regarding expansion via the male? At what point does the expansion occur through the male? And is there information available whereby treatment in a male versus a female would be different?
That's an interesting question. It kind of gets at the same question that Janetta was just talking about. So, like, I guess it goes to the basics of human reproduction, where in the male germline and the testes, you have stem cells that are constantly dividing and producing sperm. So the idea would be that this repeat is expanding during this process where there's a lot of turnover of cells. The cells are constantly dividing to produce new sperm. So to target the germline transmission of the HD mutation, would have to target those germ cells in the testes as well. So it would have to be a ubiquitous targeting. I guess we would need to test to see if the same modifiers are also working in the germline.
Thanks, Zach.
Thanks, Sarah.
Yeah, absolutely.
Because one point to note, or an implication that might be in the audience mind, is that maybe these drugs would work differently in male or female patients. We've not seen differences in somatic instability between males and females, right? So the drug targeting somatic expansions in both males and females would have exactly the same impact. It's only intergenerationally, and mainly because of the reasons that Zach just alluded to, that you see differences from generation to generation. But within somatically, so within a patient, within their different cell types, we don't see differences between male and female.
Thanks, Ricardo. So there's quite a few people that are asking about timing of CRISPR treatments or any type of treatment that you're working on. Do you think it would be beneficial for people at later stages? And do you think you would be able to reverse symptoms once people have them?
I'll have a crack at that. I think that's a difficult question for any clinical trial design. The idea is you want to go as early as possible. That's where you're going to have the most therapeutic impact. Of course, that will be difficult to achieve in earlier clinical trial phases, where you need patients to be a little bit more manifest or having a higher degree of symptoms, so that you can track the progression in a shorter period of time. That being said, we still believe that with this approach, you're essentially pressing pause on these mechanisms. So going early or going late, you're just going to preserve what you have already. So we're hopeful that even if you go late, you'll still preserve progression, right? You still prevent progression and you stabilize things. But ideally, you want to go early, pre-onset, and completely cure the disease, not have onset at all.
Agree. Thanks, Ricardo. And because I know you work on other diseases, perhaps this one to you. So it says, this is possibly a naive question, but how generalizable do you think your work will be to other diseases? Congratulations, and thank you for all your concentrated work.
I would say that we know a lot more about HD and this mechanism in HD than in other repeat expansion diseases. That being said, we're starting to learn that this mechanism is shared across these different repeat expansions. We don't know if they play a role in onset of disease, in symptom progression in other diseases. There's indications that that's the case in for the spinocerebellar ataxias, for example. Some of the genes that modify onset of Huntington's disease, they're also associated with later or earlier onset in some of the spinocerebellar ataxias, which are also caused by a CHE repeat. That's the case for XTP, which is another disease that is caused by an exonucleotide repeat expansion. So it seems to be a mechanism that drives somatic expansion across different repeats and across different diseases. So even though we don't know as much about those, I believe that this will have a therapeutic impact there as well. Maybe not to all those, but at least a subset of those disorders.
That would be amazing. So we have a lot of questions left that we didn't get to. So I will send those to our team here, and if they're willing, they'll follow up with you guys if you put your email address. If not, maybe you could email me and I could get those questions to them. So thank you all so much for your very intense interest. If you had a question that we didn't get to, I'll send those. And I want to end by again thanking everyone who has donated to the HDF to support the research that we fund, our audience, and last but certainly not least, our speakers. Thank you all so much for joining us today and sharing your work. Our next webinar is Thursday, March 21st, and it will be a data blitz style presentation of the work of 3 of our 2023 grantees, Dr. Jeff Carroll, Dr. Rachel Harding, and Dr. Tam Myrie. So you can go to our website right after this webinar ends and register. And thank you all so much for joining us, and I hope to see you next time. Thanks, everyone.
Thank you very much.